All right, we're going to talk about synapses, which are how neurons send messages to other cells. Remember from our previous lectures and readings that neurons have these components, dendrites and axons, receptive surfaces and surfaces that send messages. So the dendrites receive and the axons send. How do the axons send messages? Well, the axons send messages through changes in the membrane charge of the neuron. These changes occur and spread after they reach a threshold. So the membrane is normally negatively charged. It's more negative inside than outside. And if a threshold change, if the cell becomes less negative, that causes voltage gated sodium channels to open quickly. This allows sodium ions to diffuse into the cell, and the cell becomes even more uh, less negative and then positive. So, this is the phase 3 on the diagram. This is the depolarization phase of the action potential. The sodium channels close after a short period of time, a couple milliseconds, and then slower potassium channels open, and these allow positive ions out of the cell, and so they diffuse down their concentration, and the cell repolarizes, becomes more negative again, and it actually will become slightly more negative than it was at rest. That's the after potential phase 5 here. So once this action potential starts at one area of a neuron, it will spread to other areas of the neuron, and it will go from the nerve cell body towards the end of the neuron. At the end of the neuron, it will reach the synapse, the junction between two cells. Usually, a neuron is activating other neurons, or maybe a muscle cell or a gland cell. So, any kind of postsynaptic cell that a neuron touches will have receptors for the materials released by the neuron. We're going to go over the chemical synapses where the neuron does release a chemical. There's also some neurons that have direct contact and do electrical transmission, but there's not very many of them, and we'll kind of ignore them. So mostly neurons do these chemical signaling, where the end of the axon has an enlarged synaptic terminal or synaptic bulb that's contained. And in that bulb, there will be vesicles containing the chemical that's going to be released. It's called a neurotransmitter. At the end of the axon, there will also be voltage gated calcium channels that are crucial for the movement of the neurotransmitter. And then there's a short space, the synaptic cleft between the presynaptic cell and the postsynaptic cell. And then the postsynaptic cell membrane will always have receptors in it that bind to the neurotransmitter, and they're almost always ligand gated ion channels. All right, so the events at a chemical synapse is that when the action potential arrives, that's a depolarization followed by the repolarization, the voltage gated calcium channels will open. They'll sense that depolarization, and those cells will open. Calcium ions enter the cell. They'll bind to cytoskeleton elements, and that will cause the synaptic vesicles to move towards the cell membrane, and some of them will fuse with the cell membrane, releasing neurotransmitter molecules into the synaptic cleft. These molecules will diffuse across the cleft, and they'll bind to the ligand gated ion channels. If those channels are for sodium, when the channels open, sodium will enter the cell, and the cell will become less negative. It will depolarize, and if that reaches the threshold, that will cause an action potential in that cell. Sometimes, though, these ion channels may be for potassium or for chloride, and in that case, the cell will become more negative and an action potential will be inhibited. So we have neurotransmitters that can be excitatory causing an action potential, or inhibitory, kind of blocking them. And that depends. And so there's more than one type of neurotransmitter that can be released by a neuron. Receptor cells can have more than one type of receptor. And it turns out there's lots of different neurotransmitters. And these neurotransmitters are important, and we use them a lot. So I'm just going to kind of mention a few different ones. On this image here on the upper left, there's a neurotransmitter that we've already talked about. That shows the structure of acetylcholine. Acetylcholine is used in the central and peripheral nervous systems. It is the only neurotransmitter that's found on neurons that are activating skeletal muscle. We've already talked about a little issue with acetylcholine. In myasthenia gravis, an attack by immune cells on the receptors makes it hard to activate muscle cells because they don't have enough receptors to pass that threshold. Okay, we've also talked about acetylcholine in that it resembles, or nicotine resembles, acetylcholine and can activate receptors in the central nervous system that are responsible for mood. And so nicotine acts like acetylcholine in the central nervous system, and that's why people smoke or chew tobacco or vape, is to get that effect on the mood receptors. Okay, there on the lower left is another neurotransmitter, that's serotonin. It shows its structure there with the two rings. Serotonin, again, works in the central nervous system, and it's involved in mood. We use drugs that affect serotonin levels to treat depression. Prozac and Zoloft work not by resembling serotonin, but by blocking serotonin uptake, so that serotonin remains in the synaptic cleft longer. And for many people, we know that those things will relieve depression. Okay, serotonin's also affected in anxiety disorders. We'll use them to treat people that have anxiety, and so do things like that. Let's see here. We have dopamine that's the top one on the upper left there. Dopamine is involved again in both motor areas of the brain and in areas about mood. Dopamine is it's affected by cocaine. Cocaine blocks dopamine transporters, so thereby elevating dopamine levels in the synapse, and so affecting mood and giving prolonged feelings of elevated mood to people that take it. Dopamine is missing in people that have Parkinson's disease. It's not being secreted by neurons in motor areas, and so they can't activate their muscles. And so for people that have Parkinson's disease, we can temporarily relieve the symptoms by giving them molecules that resemble dopamine. Okay. A couple other neurotransmitters there, norepinephrine. Norepinephrine, we treat ADHD by, that's anxiety hyperactivity disorder, attention deficit hyperactivity disorders. We give them drugs that affect levels of norepinephrine. We also, I think the amphetamines are affecting those drugs as well. There's GABA down there. GABA is another neurotransmitter, gamma aminobutyric acid. It's an inhibitory neurotransmitter that can inhibit some areas of the brain. And so these, we can use these drugs for sedatives, things like that. Here, what do I want to talk about here? Alcohol affects GABA levels. There's glycine and glutamate. I think the ones I want to get to are on the last page here. Oh, yeah. Okay, the opioids. The opioids are molecules that bind to receptors for normally produced compounds called endorphins. And these endorphins block pain receptors that release by blocking the release of a pain substance. And so it can produce feelings of euphoria, but again, subject to all the problems of tolerance, dependency, and things like that. Okay, so you don't have to know all these details on these things. It's just to give you some examples and to review examples that we already know. So, in summary, synapses are junctions between neurons and postsynaptic cells, usually other neurons, but could be muscle cells. Uh, a synapse, at a synapse, the presynaptic cell will have this synaptic bulb or knob, and that will have within it vesicles for neurotransmitters. Those neurotransmitters then are going to bind to receptors on the postsynaptic cell. Receptors, that neurotransmitters can be bind to receptors that are excitatory or inhibitory. Okay, so next time we're going to start working on the anatomy of the nervous system, central and peripheral nervous system, starting chapter 13.